This lecture is going to be about lipids. Lipids, also known as fats and oils and waxes. Lipids are long-term energy storage, mostly in animals, sometimes in plants. That's why we have plant oils such as corn oil, canola oil, etc. Um, and they're concentrated energy. So there's far more energy stored in a molecule of fat than there is in a single carbohydrate. Lipids, just like carbs, are composed of carbon, hydrogen, and oxygen. But unlike carbohydrates, they are mostly long hydrocarbon chains, which means that they are nonpolar and therefore hydrophobic. Lipids are unusual in that they have family groups, and those family groups are fats, which look like this, phospholipids, steroids, and also another group called waxes, which we won't get too much into right now. But what's unusual about lipids and different from carbohydrates is that they do not form polymers. That means that we don't have a group of monomers that are linked together over and over again. But they are made of smaller couple of units. This right here is what's known as a triglyceride, and this is about as big as a fat gets. And they do not continue in a long chain. So fats, the structure of a fat, one of the families of lipids, consists of actually a sugar called a glycerol and then a long hydrocarbon chain called a fatty acid. And because of this fatty acid part, which you see here in yellow, it's mostly H's and C's, and we know that H's and C's are going to be nonpolar, hydrophobic, and therefore will not mix with water. That is why when you pour oil into water, they don't mix. And this fatty acid is joined to the sugar over here, the glycerol, by dehydration synthesis. And that's where we remove the water to join these two together, and with the help of an enzyme. So one type of fat, common fat, is what's known as a triglycerol or triglyceride. And the reason we call it a triglyceride, I bet you can guess, is because there's 1, 2, 3 fatty acids. And they're put together with something called ester linkage. I'm only introducing you to this term because you might see it on an AP, but they'll never ask you to name what is that bond. Okay? And it's the joining of a hydroxyl group from the sugar to a carboxyl group of a fatty acid. So again, put together by dehydration synthesis, and the most we ever do this is 3 times to make a triglyceride. 1, 2, 3. And with the help of an enzyme each time. Fats store energy. The longer the hydrocarbon chain, the more energy is stored, because as we talked about with carbohydrates, carbon-carbon bonds store energy. So if we look at this long chain of H's and C's, we know that this is nonpolar, which means that if it's nonpolar, it does not mix with water, meaning that if it doesn't mix with water, we call it hydrophobic. The function of fats are energy storage, highly concentrated, way more energy stored in a fat than there is in a unit of carbohydrate. But this is also meant for long term. It's much harder to get the energy out of a fat than it is to get out of a carbohydrate. All H's and C's, the more H's and C's, the more energy is stored in that molecule. About double the amount of energy in a molecule of fat than there is in a large carbohydrate. And another function of fats is to cushion organs and to insulate the body. So think whale blubber. Okay. Lots of fat on that whale keeps it nice and warm as it's floating around in the cold water. So why do humans like fatty foods? Well, we're actually programmed to like the taste of fats because they're high in energy, and so therefore we desire these fatty foods. Saturated fats. So you may have heard of saturated fat, also known as the bad fats. And they're called saturated because all the carbons have the maximum number of hydrogens that they can have, otherwise known as the fatty acid is saturated or has as many hydrogens as it possibly can. There are no double bonds between the carbons. And because of this, there's no bends in the chain, and saturated fats have really long, straight fatty acids. And because of this, these particular fats pack more densely together, which is why saturated fats tend to be solid at room temperature. Things like butter, which is almost all saturated fat, real butter, not margarine, and things like cheese. The harder the cheese, the more saturated fat there is in it. So cheddar, which is really hard cheese, has more saturated fat than, say, like a soft cheese like brie. Most animal fats are going to be saturated, solid at room temperature. And so being solid at room temperature, being densely packed together, will lead to something called cardiovascular disease. So sometimes you hear them, doctors talk about clogged arteries, or fats will clog your arteries. What they're talking about is that these fatty acids actually pack very densely together and get clogged or build plaques within the tubes of your body, which is known as your vascular tissue, which is where your blood travels throughout. So you take this small tube, small circular tube, and you start building fats inside of it, and the diameter of that tube gets smaller and smaller and has less room for the blood to flow. And eventually you can get to complete blockage, which means that the blood flow will stop flowing to your important internal organs, for example, your heart. Unsaturated fats have double bonds in it, and having these double bonds, instead of being a straight chain fatty acid, causes there to be little bends. And having the bends in these fatty acids, so having these kinks or bends, actually causes the fat molecules to be more spread out, and therefore unsaturated fats tend to be liquid at room temperature because they're going to be less densely packed. So here we have an unsaturated fat, and everywhere you see a bend, there's going to be a double bond there, and it's going to cause this bending in the fatty acids. So here's 1, 2, 3 fatty acids. So this fatty acid has 1, 2 bends, so 2 double bonds. 1 bend here, that would be 1 double bond. And then we've got more bends, 1, 2, 3, 4. So there would be a double bond at each one of those. But the big idea is that, again, this whole concept of structure equals function. The more double bonds in the fatty acid, the more bent the fatty acid, the less dense that fat will be, and the less likely that fat will be to get clogged in your artery. So we see these types of fats, these healthier or better fats. Unsaturated fats are better fats. I wouldn't necessarily call them good. We find these in plants and fishes. So they'll tell you to eat lots of fish because it's high in these omega-3 fatty acids, and those are these really good unsaturated fats. Okay, vegetable oils, liquid at room temperature, as I said before. The kinks are made by double bonds. Okay, so monounsaturated, we know that the prefix mono means 1, so that would be a fatty acid with 1 double bond. Polyunsaturated would be a fatty acid chain that had many many double bonds. But the problem with our unsaturated fats, by having this double bond, it allows something which is known as a trans fat. We talked about this before with chemistry with carbon, but around the double bond here, carbon-carbon, we have H's on the same side. We call this cis fat. This is the good fat. This is as good as fat will get. Bad fats are called trans fat, and they're called trans fat because on the double bonds in the fatty acid chain, we have hydrogens on the opposite sides of that double bond. And the problem with that is our body can't digest that properly, it doesn't process it properly, and it can cause mistakes in our cells, which can lead to cancer. And so we call it a carcinogen, which means cancer-causing agent. Okay, so saturated would be over here on the left. All straight chains, really densely put together, so solid at room temperature like butters and cheeses. And then unsaturated has these double bonds which cause the kinks, the bends, which makes them less dense. Again, this is an example of how structure relates to function. Bad fat, better fat. Another family of fats are called phospholipids. And when we talk about cell structure, we'll talk about phospholipids a lot. But phospho, hopefully you guys can guess, is referring to the fact that there is a phosphate group. And this is the same old phosphate group that you would have learned in chemistry last year. PO4 3-. And so we have our fatty acids like we had before, 1, 2 fatty acids just like we saw in the fat. But instead of having a third fatty acid chain, we replace that third fatty acid with a phosphate group. And if you notice, phosphate has a negative charge. So anything that is charged will mix with water. So we end up with a molecule that has a split personality. The phosphate end is going to be hydrophilic, and the fatty acid ends are going to be hydrophobic. Mixes with water, doesn't mix with water. So again, negatively charged. So phospholipids, are they hydrophobic or hydrophilic? Well, really, they're actually both. There's part that's hydrophobic and part that's hydrophilic. The fatty acid tails are hydrophobic down here, all H's and C's. But the phosphate group here is actually going to be hydrophilic. Attracted to water, repelled by water. And when we interact with water, it allows for these phospholipids to form barriers. So phospholipids in water, if we take one of these phospholipids and put it into water, what will happen is it forms these little spheres. And what will happen, so all these gray circles represent the phosphates, and all these little legs, these little black lines represent the fatty acids. So phospho group or phosphate group are the gray parts, and then down here the fatty acids. So the fatty acids, which are hydrophobic, will turn inward and kind of create this open space where there's no water, and the phosphate group, which is very hydrophilic, will face outward. And so what you have is that these phospholipids will assemble themselves into these little bubbles, and cells use them to form what's called a phospholipid bilayer. So this would be a monolayer because there's only one layer of phospholipid, one phospholipid all the way around, but we don't see a second layer. In cells, the membrane that surrounds the cell and separates the inside of the cell from the outside is what's known as a bilayer. So here's one layer of phospholipids, here's the other layer of phospholipids, a bilayer. Water on the outside, water on the other side. No water in between. So why is this important? Phospholipids create barriers in water and define inside of a cell from outside. They make cell membranes. We'll come back to this when we talk about cell membranes later. Another class that's really important is something called steroids. I'm sure you've heard of steroids before. It's very controversial in athletics. But steroids are actually referring to a group of fats that have this ring structure, 1, 2, 3, 4 rings, this basic structure. And when we did the chemistry of carbon lecture, you actually saw these when we talked about testosterone and estrogen. So we have 4 fused carbon rings, and by adding different functional groups, we get different functions of the steroids. So examples of steroids are going to be things like cholesterol and sex hormones. So estrogen and testosterone would be sex hormones. This right here happens to be a cholesterol. Cholesterol is an important component of cell membranes. They actually allow cell membranes to be flexible. They're a precursor of all of their steroids. So we use cholesterol to build any other steroid. So meaning estrogen is actually built from cholesterol. Testosterone is built from cholesterol. So high levels in the blood, though, can contribute to cardiovascular disease. And we'll talk about this a little bit more later. Again, cholesterol is an important component of cell membranes. It helps to make the cell membrane flexible. So here are phospholipids, and here's a cholesterol here. And what it does is the cholesterol actually breaks up the interactions between these phospholipids, allowing there to be more wiggle room. Helps keeps the membranes fluid and flexible. Again, looking at steroids, all of these are steroids. And the only thing that's different is the functional groups attached to these carbon rings. And so we can have things like cortisol or testosterone or estrogen or progesterone or aldosterone. All of these are actually cholesterol with different functional groups. Okay, so that is our lecture on lipids. I hope you enjoyed.